So we're going to have a look at this characterization of innate immune responses to AS02 adjuvant H5N1 vaccine compared to non-adjuvant H5N1 vaccine. This has 898 samples, so it's a very decent sized data set. If I go to the downloads, there are cell files as well as the matrix data. So it's already been processed, or you can get the raw data file, which is always nice. The raw data file is very large because there's 898 image files, which is 4 gigabytes worth of data. There's a README CSV file, which is particularly important. You need to click on that one and download it. So we'll grab that and open it to see what's happening within the particular set of data, because this is a fairly complicated data set, and we can do an analysis with GEO2R, which is definitely good news. So if we take a look at that CSV file, so I'm going to open in so now I've got it downloaded. I want to open it in, where is it? What's it called? It's called GSC whatever. So that file is this CSV file. I'm going to open with that Notepad++ because that's my default in this particular case. And what have we got? We have a file which has 44 numbered subjects, but if you look, you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27 is missing, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41's missing, 42, 43, 44. I think that's just 2 numbers missing out of the set. To be honest, let's open this again in Excel, or let's open Excel and then open it with Excel. So let's go here, grab Excel. Excel's here, and I'm going to open a file and browse it from my local directory. Go to downloads, wherever downloads is, downloads, and where's the CSV file? All Excel files. I want all files then. The CSV file, open. That's not at all helpful. It didn't do a proper import, which is what it should have done. Well, exceptionally annoying. Because I'd really prefer to look at it in actually column formatted so that I can see it properly. Why is Excel not doing things sensibly? Wait a minute, there must be a way of doing it properly. In Excel, open, browse. Can't I do an import? Ah, cancel. And it says .csv. It should automatically do an import on it and then allow me to change its shape and structure. It's because I've opened it. What? Here's Excel. So why is it doing that? File, info. Why? What other programs can I open it with? I can open it with OpenOffice, I suppose. Is that going to behave better? Open with OpenOffice Calc. Is that going to open it properly? Yes. Thank you. Nicely. I accept. Right. Now, this is an easy way of looking up the thing. I'm not quite sure. Okay. I do get why they did that particular thing. That setup. So you've got a space there, and then you've got a thing saying demographics. So the demographics goes across all of those headings. So the demographics is their gender, their age, their ethnicity, and you've got the vaccination time frame, so when they were vaccinated. So this study is from 2018. And then you've got whether they had the adjuvant or not. So what you've got is the particular subjects that have had the adjuvant. Let's give them a colour. Edit menu. Oh, I just want to colour the thing. I don't want to do revision automatic. I don't want herramientas. Why? It's I just want to colour that line. Why? I don't want to insert anything. I don't want to move anything. I just want to highlight the ones that say yes on it by changing them to be a different colour. Here. Fine. So I can put these in yellow. Great. I'll put them in the same one. Why can't it just default to Amarillo and me just click it? Amarillo. Is that working on Amarillo? Fine. Right, so have I missed any? It's kind of important to get it right because this is a phenomenally complicated data set. I'm going to save this as an Excel file or some other format once I've got this formatted so that I don't lose all the colouring again. So I can see. I could have done the 3 in a row. Idiot me. There's 2 in a row here. Could have done it by conditional formatting, but that takes longer to think about. Right, so those are all the yes cases, so I needed to put that in mind. I'm going to save this one, and I'm not going to do it as a CSV. I'm going to do it as an Excel file, and the only type that I can do is an Excel Excel 95, no. Excel 5, no. Excel 97 2000, whatever the Excel is. That's close enough. It'll have to do. So we'll do it as that. Excel will open it, but it won't open it with a great amount of happiness. It's also saved it as open ODS. Okay, then do it as an ODS. I don't really care. I just want a saved file. So what's it done? Just saved the ODS? Yeah. Open with, if I open it with Excel, does it open it properly? It's locked for reading. Oh, open it read only, that's fine. I haven't got any calculation settings, that's all perfectly fine. Fine, I can read it in both. And I don't need it there. So, what do we need to do? We need patient 1, 2, 5, 10, 11, 12, 17, 18, 19, 21, 23, 25, 26, 28, 32, 34, 38, 40 and 43. Need to be in our yes set. Now, I did this one before in terms of defining groups, but it's a little bit of a tricky data set. So let's get it as it was done originally. So this is in the original order that they were done. Is the original order on titles? Let's do source now. Right, they're sorted now by individual. This is patient 1, patient 2, and so on. Now, let's see what they've done. Here is the accession number. So each of the slides, each of the samples that was collected has these accession numbers. So you can get this, and this will give you an image of a single microarray slide. This is the code that they've given to it. So it's the H5N1, individual 1, day 0, after 12 hours, and then it's 14B01 is the sample that they were given. So because this is going to be a blinded kind of test, the person's not going to know if it's 14B01 happens to be a vaccine with the adjuvant or without the adjuvant, because they're going to look the same. So if I go through here, it says individual this, and it says time point. And then you've got, the way this study was done was they were treated, then you took samples of them. So this is the sample before they were treated with the vaccine at all. This is them once they're treated with the vaccine after 12 hours. This is after 1 day. This is after 21 days, 21 days and 12 hours, 22 days, 100. So that's the kind of infinite time thing. Then you've got a booster that is given. I can't remember when the boosters were given. It must be 3 months, because otherwise 100 days doesn't make any kind of sense. And to take it must be even after 4 months. But anyway, they've got a booster. So this is the 0 when they're on the booster day. For some unknown reason, this person's been measured twice. No idea why. They've got that particular thing. But they have. So you've got 2 different labels on this for no apparent reason. You've got 1 at 2 hours, 1 at 4 hours, 1 at 14 hours, 1 at 12 hours, 1 day, 7 days, 21, days. "21 days 2 hours, 21 days 4 hours, 21 days 12 hours." So in the 21 days they were there for the whole day in the lab being plotted and tested. 22 days, 28 days, 42 days, 100 days. And then you've got the next patient. So what you've got is patients, about 42 of them. You've got time points, many different ones. You've got first and second vaccinations. And there's a sort of, I'm not sure they have exactly the same data collection schedule of each of them. So let's look at the second person. That's the first person. Second person, no, they don't have 2D zeros on the second day, do they? No, they do. They were still collected twice. That one. So it looks like the time periods might be exactly the same for all patients. So what you need to do is count how many is in the time series. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Make this full size. And start again because I've lost count. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 was to the DO1. 14, 15, 16, 17, 18, 19, 20, 21, 22. So there are 22 time points for each one of the patients, supposedly. 22 times 42. No, just do a plain 22 times 42 is 924. We've got 898, which tells me that they've messed up some of the calculations. So that means some of the things are missing, and you can't just take the file and guess which ones are, well, not guess, you can't just work it out based on pattern order which ones are which ones, which is not ideal, to put it mildly. To be honest, can I download this spreadsheet, this CSV? Can I download this table? Because if I could download this entire table, I'd be a much happier bunny. Anyway, what I'm going to do is define groups, which I'm going to get rid of these two groups because I'm going to start again. Damn, I should have kept those groups because they had some interesting results. No, they didn't have any interesting results. So what I'm going to do is think of the possible combinations and experiments they can do. So what I'm going to get is one group which are going to be all of the treated people on their first vaccination, so not having the booster, after 12 hours. So I'm going to define that as a set. So I'm going to call it first and 12 hours. So that's the group name that I'm going to call that. We actually need to change that. First 12 hours and treated. So I'm also going to get a first 12 hours and untreated group. Right, so which ones were treated? So this one is in the first 12 hours treated. So if I have my spreadsheet, which happens to be in my other window, but I can remember, it is person 1. And I'm going to sort these columns by time point. So now, why are they on sort of random numbers? That's not at all useful. That's really a pain in the neck. Okay, let's keep it ordered by the individuals, because otherwise there's a chance of me making a mistake. So this is the 12 hours which is there after their first vaccination. This is the 12 hours after their second vaccination. Now, there's nothing in their names here to say this is first and this is second. This is very annoying. The only way of me sort of telling is because of the order they are in this particular list. So this is very annoying, because it would have been a lot more intelligent of the people to put something in here to suggest whether it was from the first or the second collection. So now I have to hope that they've put the data in this folder in the right sets of orders. So now I need to go down to individual 2. So individual 2, I need to find their first hours, and I need to add that to the sample. So they're treated. I can create another group, which is the first 12 hours and untreated. So I might as well do both groups at the same time, so I don't have to go through the list twice. So the third person, when I get down to individual 03, this one here, this is untreated. And I need to go to the fourth, which is also untreated. The fifth, which is treated. The sixth, seventh and eighth are all untreated. So sixth, seventh, and eighth. All untreated. Oh, and ninth is also untreated. These were assigned randomly, which is why they don't look random. Then 10th, 11th and 12th are all treated. And 13, 14, 15, 16 are untreated. Am I just worried if I ticked the wrong box for 13? I didn't. 13, 14, 15, 16 are all untreated. 17's treated. Eighteen is untreated. 19 is treated. 20 is untreated. $21 is treated. $22 is untreated. $23 is treated. $24 is untreated. And 25, 6 and 8 are all treated. And 27 is missing. $29 and $30 are not treated. $31 and $2 are treated. $31 and $32 are treated. $33 is untreated. $34 is treated. Oops, let's get the right one. 34 is treated. 35, 6 and 7 are not. $38 is. What? 38 is the one that's had a completely different pattern of checking. So this person, they did at 2, 4, they did twice at 0. So this is like the scheme that they did for the second test. So let's go back and have a little look if 38. $38 has only got one set of measures. And this is the measuring pattern of the second one. So it looks like they forgot to measure person 38 in the first regime. So they forgot, or they lost, or something happened to their samples in the first case. So I'm going to take that one out. or I can leave it in and see if that's exactly what I thought. I want that one. Yeah. I'm going to take 38 out because I don't trust that data. I'm pretty sure that's the second after the boosting that they collected it, and that's why there's missing ones. There's not 924, there's only 898 because they missed that set of data. They've got to have missed it on another person as well. So 38 is not there. 39 they didn't, is a non. 39 is untreated. 40 is treated. And 40 has that same problem. Yeah. So 38 and 40, they only collected half the data. 42 is untreated. 43 is treated. And 44 is untreated. And 44 again, you've only got the pattern of the second collection. You haven't got the first collection. So that explains the missing data. So the case, so that I need to note down somewhere. So I'm going to write it in the. I'm going to add a column to the spreadsheet, which is going to say missing the first. So I'm just going to put Y's for the ones that are missing. So 38 is missing. 44 is missing. And which was the other one we said was missing? 44 is missing. 43 is complete. 42 and 40 is missing. So the ones that are missing are 2 that had the adjuvant and 1 that misses the adjuvant, and they didn't do the first, didn't do any of them collections on the first one. I don't know what they did wrong. What they did in that particular thing. Yeah. It's also, I'm not sure about the gender balance in the thing. Right, we've created our data sets and I'm pretty sure those are correct. So now I've created 2 sets. I can compare those to each other. So I can compare which genes are differentially expressed between the 2 by click the analyze button. So it will go and do the processing and analysis of this particular thing. Using this web tool is by far the easiest way of doing it, but it also will give you an R script, which we can look at here, which allows you to import it into R, and we can do a more detailed example. So it allows us to move from this particular case to more advanced cases. Usually it doesn't take quite that long. Okay, done. So the nice thing about this, it's not what I kind of expected. Okay, so let's go through the graphs first. This thing is called a volcano plot. What it shows is the log 2 fold change along the x-axis. So that is how much, as a multiplier, the amount of gene expression changes between the treated versus the untreated group. So I've done it actually the wrong way around. It doesn't really matter. So you imagine the treated going to the untreated. So is there twice as much in the untreated group, in which case it would measure 1 on here. If there's 4 times as much, it measures 2. If there's 16 times as much, it measures 4. If there's a quarter as much, but actually, so if it's double, it will go to 1. If it's 4 times, it will go to 2. So this is along this x-axis. If it's half as much, it goes to -1. If it's a quarter as much, it will go to -4. So almost all of this is on the positive thing. So there's more of it in the untreated than there is on the treated. Now the other thing it's done is the minus log 10 of the p-value. So this is the p-value for is there a significant difference. So you want the minus log 10s to be above about 4 point something. So the logarithm which turns 0.05 into being significant. So they're actually coloured for you to show which ones are significant. So all these blue ones, these ones are down regulated. So 3 genes are down regulated between treated and untreated, and all of these red ones are up regulated. So if I had it the other way around and I had untreated first and treated second, these would all be down regulated, these 3 would be up regulated, just the way I plotted the thing. That's the volcano plot. Here's the same information, so the log 2-fold change, and this is the log 2 of the amount of expression that there is. So some genes might have 50 copies, so 2-fold change goes from 50 to 100. Some you might have 1,000 copies, so 2-fold change goes from 1,000 to 2,000. That's a much bigger change in the absolute amount of things, 1,000 as opposed to 50, but is it more significant or not? So this is comparing the log of the absolute amount of the expression of things. So here, because it's all in base 2, here you've got 16 units, here you've got 32, no, you've not got 32, you've got 64 units, here you've got 256 units, here is going to be 1024 units. So they're all in powers of 2. You don't really need to know very much about that, apart from the genes that are in the red. So upregulation, some of them are low levels, some of them are high levels, they're all over the place. The ones that are down regulated tend to have more copies than the others, and there's no particular exciting thing. This thing over here got a massive fold change, but it's an incredibly variable gene. So because it's very variable between the 2 groups, it's not actually come out as significant. Now this is the thing that when I opened up the results and I just looked at it, I was going, whoa, that's a bit weird. What you would expect to happen is you would expect all of the cases to group together by colour. So you'd expect all the purple ones to form a cluster and all the green ones to form a cluster, showing that they have the same sort of expression to each other. So all the ones that have had the adjuvant would behave in the same way, and all the ones that haven't had it would behave in the same way. Now when I first did this and I mixed together all of the so the 12-hour from the initial giving of the vaccine and from the booster of the vaccine, I got 2 distinct groups. So one down in this part corner and one up in this corner, but they were a mixture of purple and green spots, and I was like, what does that mean? So there is a very significant and very clear difference in which genes are switched on just after vaccination and after boosting. There is a completely different pattern of gene expression. So that is a definite significant thing. And it happens, the same differences happen whether you've used the adjuvant or if you've not, which is why the purples and greens are mixed together. This case, because they're all from the correct and same time point, there is no obvious clustering based on this thing, which is a bit odd because that would suggest that your adjuvant is not doing anything particularly interesting. It's not having a very big change. This is how many genes are significantly different between the two. So out of a total of 53,617, 43 are different, which is fine. You don't really want a massive amount of differences. This plot is called is a plot to check that the samples are all equal to each other. Now, when you create an experiment, when you're doing repeats of an experiment, you might put more or less sample onto the thing. So the first job you have to do when you're comparing all your samples is to make sure that they all have the same basic level. They're all standardized to having the same concentration across all the experiments. So this process is called normalization, and it involves you getting the box plot of the amount of expression and making sure that the medians and the interquartile ranges pretty much align with each other. And you do this just by mathematical fiddling around to make sure that they're all stacked up over each other. So it's well normalized and the data is pretty reliable. You should also see the same sort of pattern and shape in the intensity on the images that you get of their expression. You shouldn't have any which are very different to the others. That would suggest that somebody's used the light scanner for reading the data incorrectly, or they put their fingers on it, and it just suggests a worrying data point. So here there's also no distinguishing features between the treated and the untreated group, which would also be very worrying. So that's all good. Says your quality control's fine. Here's the p-values. They form a nice exponential kind of distribution. There's lots at 1, and then they've got a gradual increase from very low values. That all looks good. If it's all at 1 and there's very little else, then it's a bit odd. This is the values of your t statistic. It forms pretty much on a straight line, so that's also good. You want a bit of variation at the top and bottom because those are the unusual genes. I don't see there's anything particular that you want to look at the mean variance. There's a line through the middle. They should be roughly scattered around it. You worry about things that sit up here because they're very different to what you expect, but otherwise the shape's fine. And here is the table of differential genes with their p-values. P-values, the calculated t statistic, the log fold change. So this one is a 2-fold upregulation. This is nearly a 2-fold upregulation. So all of these are up. This one is down to being half. Down to, no, not quite to being half, to being 1 over root 2, but anyway. Then it tells you this is the database access number for the GB database, and these ENST numbers are the ensemble codes. So these are the annotations in the human genome, and these are just the codes that are used by the supplier of the microarray to label the oligonucleotides of those particular things. So if they're not associated here with a particular thing which is in the database, then it means these are oligonucleotides which are known to be within a conserved part of the genome, but we don't actually know what they're doing. So you can look these up and find what they are, but they're not well defined, whereas these things are. So the only real thing that's got the top thing that's got a very clear thing is this ENST, whatever it is. So you can look that up and find out what it is. So you want to look at all of them that go down to have a p-value that is less than 0.05, which is down to here. Now you can download this table. If I click on download the table, it will hopefully download things. Fine. while that's doing that, you can go back and look at this. There are other options you could do. So you can change the way the p-values are detected. You can change thresholds and things to make your plots look different. And here is the important bit, which is the R file. So this is code that will run in R to do that analysis. Now I'm going to copy this code because I happen to have an R window here, and I am going to take the existing version of the R code that I've got, which is a bit wrong, and I'm going to paste it there. So hopefully that's right. Okay. Nice. That is fairly nice. So we can look at it easier on this screen. So remember I was going through and setting up the data sets, and I had to pick which one was in which group. So it is done using this particular system. So each of these X's is an experiment that or a sample that will be excluded. So the way the data set was set up is you have the samples after the initial vaccination, and then you have the samples after the second vaccination, which are in this second half of the this matrix of X's. So this is saying skip the first one, which we know is the patient with before they've actually been vaccinated. Collect the one after 12 hours, and that one is in group 1, that one's in group 1, this one's in the untreated group, which is 0. This is in group 1, this is in group 1, this is in group 0, this is in group 1, this is in group 0, and so it goes on and on to do all that. Okay? So this is a useful thing, because when we if we decide, oh, I'd like to do a different analysis, then we can just change what we've got in this particular collection of X's, and we'll end up with a different analysis. So you can either do it by going through the samples, going through this sample list and set, and defining which one's which is what I did by hand, or you can do it by changing this R script. So doing it by selecting them out of the web interface we can't do when we do it on our own computer using R. So now I know the structure of what this means, it makes it slightly easier to figure out which samples and which ones I want to put into my data set and which ones I don't. So that's sort of nice, though it's still an absolute pig. That's the worst looking system I've ever seen in my life, but at least I get an idea about how that's set up, and I can possibly automate a system to create sets within this data, which will make my life easier. Anyway, I've got that saved for later. I can repeat exactly that same analysis using R just by running that set of script. So if I run that, it will give me all of these output graphs and things, the results. It'll give me all of this in R itself, which is better because I'll get higher quality images than I've got here, and I can then put them into my final documents. So that's the basic analysis. Has it? No, it's downloading everything. I just want really the top scores. Really. I clicked download full table. I shouldn't have done that, because it's going to do all 53,000 lines. Please. I just want to download the important ones. Options. Reanalyze. No, I don't want to do that. I just want to download the important top genes. There must be a way of downloading just the ones you want. If you click on it, you get to see the differences. So this is what levels that particular gene is in treated, and this is what level it is in untreated. So here you're saying that it's higher in the treated group and lower in the untreated group. Though you can see that some people, like this one, is higher than this one. So particular individuals don't behave like absolutely everybody. I'm quite surprised. Is that download? Really? Time running is 31 seconds. It feels a lot longer than 31 seconds to me personally. So there's my fun sample list that I was going through. Even worse, when they're listed in this way, they're not even listed in the patient order properly. Like, really, what on earth are you doing? And that's important. Oh, I really... Bro, that's so horrible. That is seriously horrible. So if you were so what I was thinking about was going to process the raw data and do the analysis again from the start. So not use their processed numbers. And it would be absolutely a nightmare to do, because they're not even stored in the correct order. You can download a list of accessions here. Okay, let's have a look at that text to see if it is actually correct. Okay, that tells you all of the accession numbers, but it doesn't tell me the patients. Oh, that's such a nightmare. This is an exceptionally annoying dataset to deal with. It has finished my download. Yeah. Oh no. No, that's not that one. It's finished the top table. So here is the table that I wanted. Remember, it's done all 53,000. When I said I only needed the top ones that went down to being a significant p-value. So the p-values are significant down to 0.05. You want the adjusted p-value, which is this column. So they're validated down to the first 43 genes, 42 genes, because there's some labels at the top. Right, so this lot above are the ones that are important, all the others are not. Right. So that's a complete set of data. What was this? Query datasets. Okay, so you could pull out individual datasets if you want to. So you can download individual cell files. I wonder if you can see a cell file. Let's download a cell file and see whether I can see a cell file. So it's downloaded the cell file. And it's downloaded a cell file, and it says a cell file is an image file, which is what it actually is. But if I open it using GIMP, is it going to show me what the cell file looks like? It's going to be interesting if it does. Yeah, where's the file? Can't see anything. Do you see anything? Nope. Let's go back a minute. Let's go to downloads. This is the I want to extract this file. So I extract the file so that it becomes so I get a cell file. That's exactly what I want. Now I want to try opening it in GIMP. And does it open? No, I can't see anything. There's nothing in the image window. Open with. No. What you should see is a file which has loads of dots on it. It doesn't usually matter. Because I've downloaded all of the cell files. And I'm going to then need to sort that out. Okay. So that's definitely